Hi everyone. Time flies, right? It's already December 2021. It's me again, Stella, USP Strategic Customer Development Manager for Southeast Asia. So as usual, before we start, a couple of housekeeping information. Now, if you cannot hear us at this time, please follow the audio instruction to set it up. Otherwise, you can click the session information and you can dial in to listen to today's session. Next, if you have any questions that you would like us to address live, please feel free to key them into the Q&A tab on the right-hand portion of your computer screen, and please do make sure that your question is sent to all panelists so that our speaker for today, Dr. Christian Zeiner, will be able to see your question and address them. So with that, we are all set for today's session. Now, good morning, good afternoon, good evening to everyone from wherever you are. Welcome to our December edition of the Quality Hour, the last edition for the year. Quality Hour is a series of webinars developed by the team here in USP Asia Pacific to stay connected with you. So for today's session, we will have Dr. Christian Zeiner to discuss about the value of pharmacopoeia reference standard. And then I will give you a quick update from USP and some of the upcoming activities in January 2022. So before we end the session, we will have some time to take on some questions. So again, please feel free to type in any questions that you may have and you would like us to address live in the Q&A tab. We will do our best to address as many questions as possible. Otherwise, we will always follow up with you offline your questions. So without further ado, I will hand over the time to Dr. Christian Zeiner, Senior Manager, Scientific Affairs, Europe, Middle East, and Africa region. He has more than 17 years experience in the field of pharmaceutical reference standards with the focus on small molecules. Today, he will be sharing with you an overview on how pharmacopoeia reference standards save your work and increase your efficiency in your laboratory. So over to you, Christian.
Thank you, Stella. Let me share my screen here. So I hope you can see my slides.
Yep.
Okay, so thank you very much, Stella, for the introduction, and good morning from my home office here in Germany. My name is, as Stella said, Christian Zeiner. I am the Scientific Affairs Manager for the EMEA region, and I'm very happy to be with you today on these Apec Quality Hour here. I normally work from our USP office in Switzerland, in Basel, but right now, as I said, from the home office in Germany. During the pandemic, we are still in home office. So it's about the value of pharmacopoeia reference standard, as Stella mentioned already, and our agenda will be that I will talk quickly about the USP, then we will have a look on what conclusive results are and what risk you are accepting or maybe not even aware of when you are trusting inconclusive results, and we will have a look what these inconclusive results are. And then we will look on the risk from the point of uncertainty, which is also an interesting standpoint and opens a new perspective that was not so far, or probably not so far, thought of with users of reference standards, secondary reference standards, in-house and working standards, and a bit of conclusions as well. But let me start with a small introduction to USP. When we talk about USP, we are talking about more than 200 years now of building trust. Back in 1820, when USP was founded, it was a single recipe book, and now 200 years later, It's not a recipe book anymore, but much more about procedures and acceptance criteria to support medicinal articles in the marketplace. And we do that with more than 4,900 monographs nowadays, with specifications for identity, for the strength, especially for the quality, which is our main task to assure, purity and packaging as well, and also labeling for substances and for dosage forms. And those 4,900 monographs, they are also supported by more than 350 general chapters that are providing also clear step-by-step guidances for assays, tests, and how procedures should be performed. USP was first mentioned in law in 1848, 28 years after the foundation of USP, in the Drug Importation Act. That act required imported drugs to meet the standards for strength and purity that were established in the United States back then, and in the USP, of course, then. And that legislation helped to solidify USP status as a national compendia and set also a precedent for future federal drug laws. And the next drug law was set up as the Food and Drugs Act in 1907, following a scandal in the United States where the book In the Jungle was published by a writer called Upton Sinclair, who was describing the practices in the meat industry back then. And at the same time, so that was so realistically written and was so much based on facts that there was an outcry in the public. And it really happens that then the Food and Drugs Act was set up. The scandals were all in the meat industry, but at the same time, nonetheless, then the law in the drug regulatory area was set up there as well. And then the next law was set up in 1938, the Food, Drug and Cosmetic Act, again following a scandal that happened in 1937 when there was the medicine elixir was contaminated or adulterated with poison solvent diethylene glycol, and during that scandal 107 persons were killed, many of whom were children, and as a consequence the Food, Drug and Cosmetic Act was set up. And that Food, Drug and Cosmetic Act is still in place in the United States. There were of course different versions set up, and here you can see from the current version that when it comes to naming and identity, to packaging and labeling, to compounding here, but especially to strength, purity and quality, then it is always referring to the official compendium, to the USP here. And you can see it here, especially for the strength, purity and quality. And what does that mean in practice? Every medicine that is brought to the United States market, for example, needs to fulfill, whenever there's a monograph for that drug substance and then also that drug preparation, whenever a monograph like that is available in the USP, then the drug needs to, the medicine needs to comply to that monograph, to the specifications that are set there. That is the level of quality that must be fulfilled in the United States when a medicine is mentioned in the USP. And it does not even need to be written on the medicine that it is compliant to USP. It could also be left out, but even then it is necessary to fulfill the specifications. And we will come to that in a second once again when we talk about conclusive results. And that is the case in the USA, of course, for which USP was originally designed, but a lot of other countries also acknowledge the quality standards of USP in their drug regulatory framework, and the same would apply there, of course, as well. Excuse me for a second. So, and we do give that quality standards by setting up what we call compendium standards. These are the monographs and the methods that are written in the monographs, the methods that are also written in the general chapters and all as well. of course, and that is what we call compendial standards. And these compendial standards, they are data-driven and grounded in science by a transparent setup process. What is that transparent setup process? Well, before a monograph, a compendial standard, also general chapter, becomes official, it's published as a draft in what we call the Pharmacopeal Forum, the PF, and user stakeholders all over the world have free access to the online version of the Pharmacopeal Forum. You can set yourself their alerts, for example, when you want to be notified when a certain monograph, for example, is happening to get a new draft. Then you can look into the PF and see that draft, can see if that draft is applicable on your products, for example, and you can also send them comments in a 19 days period. Where we collect comments, where we then check all these comments and view them on their relevance, assess them on their relevance, and sometimes or very often put these comments in a way that we change the monograph, for example, and only then is the official monograph then published in the USP, and 6 months later it's then becoming also official. And these compendial standards, they are used by manufacturers and regulatory bodies to help ensure the quality of pharmaceutical products, and they also facilitate significantly the access to affordable medicines. That was found out in a paper that Irene Moreno-Westel and her co-workers set up here. You can have access to that one as well for free on the internet. It's an open access publication here, and also the compendial standards was found in a study of my colleague Ian Worthen and also other colleagues of USP in a study there with doing surveys. It was found out that compendial standards are also of a high value to the pharmaceutical industry, because the pharmaceutical industry thinks they are decreasing the development effort of pharmaceutical articles as well. So these two publications, these two papers are available and are underpinning how USP and also other compendia, of course, would probably support the market, the pharmaceutical market for quality, for example. This was also found in a white paper from the International Meeting of World Pharmacopoeias, that is under the umbrella of the World Health Organization. And there the World Pharmacopoeias are meeting, and they also published that white paper there, where it's also saying as a basic message that pharmacopoeial standards facilitate the preparation and the assessment of regulatory submission of the dossier that you are submitting to get your marketing authorization in a certain region. So that's where pharmacopoeial standards, compendial standards help facilitating, and also compendial standards are providing there a public mechanism for an independent verification of the quality of a product, and that at any time during its shelf life, because that is what is very important. You have to fulfill with your medicine, you have to fulfill the specifications of compendial standards during the whole shelf life. So not just at the point of release, but no, during the whole shelf life, to make sure that the risk for the patient is, of course, also as low as possible. So that's basically about compendial standards here. Let's have a look now on conclusive results and what these conclusive results then really are. Before we come to that, let's have here the starting point again, compendial standards that we have just discussed already before, and we have said that in order to ensure the safety and the efficacy of active pharmaceutical ingredients, of APIs, and also their finished dosage forms, of course, that these pharmaceutical articles need to be of a certain quality. And the USP and other pharmacopoeias, for example, they establish these compendial standards to ensure the quality requirements. That's what we discussed briefly also on the slides before. And we also mentioned already that they are set up in a very transparent process. What we need to mention now here is that they consist normally of two, let's say, types of standards. So we have the compendial standard, we have the documentary standard in there. For example, the general chapters and the monographs and the methods in there. And then also we have in these methods described the physical reference standards. And these physical reference standards, these pharmacopoeia reference standards, compendial reference standards, they are also sometimes called. They are almost always primary reference standards, first of all. So primary reference standard is when you go to the Q7 guideline of ICH, you will see there that the definition is saying that it is set up by an analytical process without any reference to another standard of the same kind, for example. So it's an independently set up primary standard. And this primary standard is then established also by a robust collaborative approach, and it's assuming official status when it is connected to the documentary standard, to the official monograph or the official general chapter. That's also outlined in our USP General Chapter 11. It's mentioned there to a certain extent as well. So let me elaborate a little bit on the robust approach. What is happening there when the material is testing in a collaborative way? So we have colleagues in USP, reference standard scientists, they are called, and they are setting up for a certain reference standard a collaborative testing protocol. It's designed by them and then approved by their peers and also their managers. And for example, when a reference standard is used in the monographs, for example, for the assay test, then this value is assigned by a process that is involving at least 3 different laboratories that are checking the reference standard due to that work protocol. And using that collaborative testing, each laboratory is then generating data by the tests from the work protocol, reviewing that data as well, and sending that data then to the reference standard scientists. And they are checking then on agreement between the tests from one laboratory. Does it make sense what one laboratory is sending to us? The laboratory themselves, they have also already checked, as I said, they reviewed that data as well. But nonetheless, the reference standard scientists are looking once again on the data from one laboratory and check, does it make sense? Does it add up all to 100%, or are there any data that are contradicting each other? So once that lab is kind of checked, the tests are checked, then all the laboratories, the results there are also compared against each other. That does these data make sense then between the laboratories, or are there outliers, for example, from one laboratory? And once that is ticked off and checked there, we use then normally for quantitative standards the mass balance approach to assign the label value then for the reference standard. And the data scientists, the reference standard scientists, they also generate the label text. That is all then in the end collated in what we are calling a reference standard candidate evaluation package. And that package with all the data is then presented to our expert volunteers so that they can review that data once again as well. And they, as the volunteers, so external people to USP, it's not USP staff that is making that decision, but external people. That is that very transparent process I was also talking of before. So the volunteers, external people to USP, our volunteers, they are in the expert committees, in the expert panels. They are checking the data and finally balloting on the release of that reference standards. And that whole transparent process and robust process with 3 laboratories. That is very important in order to make sure that when we have the official status, we are really the point of reference, the compendial standard with a reference standard inclusively is building really a point of reference on which pharmaceutical companies can then rely. And that's also why reference standards, but why compendial standards and the combination of the documentary standard and the reference standard within is thought to be conclusive, is assumed to be conclusive and authoritative. That is, they are assuming really that official status there. And it's mentioned, for example, in the European Pharmacopoeia in their general text 5.12, where the European Pharmacopoeia is saying that whenever a reference standard is referred to in a monograph or a general chapter, then it's representing the official standard that is all alone authoritative in the case of a doubt, for example. And with slightly different words, it's also like that in our USP General Notices 5.80, where we are saying when the test in the USP or in the national formulary calls for the use of the USP reference standard, then only the results from that method together with the specified USP reference standards are conclusive. And what does it mean in practice? Well, it means, and we mentioned already the shelf life before, that it also needs to fulfill, the medicine needs to fulfill the specifications during its whole shelf life. So it could be, for example, that the regulatory authorities in order to check on the quality in the market, go to a pharmacy, go to a drugstore, they pick products from the shelf, they know what compendial standard is relevant for that specific drug preparation then, and they check the specifications of that medicine, or the specifications of the compendial standard on that medicine with the combination, with the official or authoritative or conclusive combination of the method from the documentary standard and in combination with the reference standard. So that combination is then the benchmark, and only this combination. And in consequence, it means then also that other combinations of the documentary standard and the reference standard would not be conclusive and may potentially increase the risk here. So, for example, you use the documentary standard and you use an in-house standard that was compared to the official reference standard, for example. That combination, even when it was compared to the reference standard there, is in last consequence not conclusive. It could always be that the regulatory authorities are using the official combination and come to a different result, and that is where the risk is lying. The same is when you use the documentary standard and a non-compendial standard from somewhere else, maybe secondary or primary from somewhere else. It's also not a conclusive combination and may also potentially increase the risk. So to show compliance to the USP, the only conclusive combination is the method from the USP and together used with the reference standard from USP. So, and what is this when it comes to secondary standards, to working standards, to in-house standards that are also secondary standards compared to the USP? That is not only introducing risk by the non-conclusive results, but also by a bit of uncertainty that they are bringing into the game, and that are also in addition increasing the risk here. And the next slides are about that aspect and how uncertainty is increasing risk here. So to understand uncertainty and risk, we should have a look on the measurement hierarchy. And in our brochure, not all standards are created equal from last year. You can download it also from the website with the link below here. there is the measurement hierarchy triangle mentioned. And on the top of that measurement hierarchy triangle, we have the defined unit of measurement. That could be an SI unit, for example. It could also be another unit derived from a combination of different SI units. In the case of a quality test, it is of course something like a purity, a potency, a concentration, for example, like what we have on our label, milligram per milligram of material. So on our label, for example, says this reference standard has a potency of 0.997 milligram per milligram of material. That is what is on the label of our reference standards when they are quantitatively used. So that defined unit of measurement stands on the top of the measurement triangle, and we have then a primary reference measurement procedure like the collaborative testing that is assigning then the value to the USP reference standard by what I just explained a bit more in detail here. And then the USP reference standard would be used together with the USP compendial procedure, normally in that conclusive combination, to assign the value, the reportable result, the reportable value directly to the manufacturer's product sample, like the ibuprofen tablets, for example, or any other medicine. That is the conclusive combination that we were talking about. But you also know that very, very often manufacturers in our standards, our working standards, are put in between, and that is normally done by a comparison to the USP reference standard or other compendial standards by the compendial procedure. And only with that one then is maybe again the compendial procedure used in order to assign a value here. So you have then in the end then the results as at the bottom. And you can see, however, here on the right side that every bit of these steps is introducing again uncertainty here. That principle of the measurement triangle is adopted from the ISO guide 17511. But that is a basic principle of measurement. Every measurement step, every second step that you put in is of course increasing the uncertainty here. And so it is definitely clear that the conclusive combination is providing the least uncertainty to your result. It's the conclusive result, but at the same time it's also providing the least uncertainty. And every step that you put in there, a secondary in-house standard or working standard or how you call it, that is of course introducing additional uncertainty here. And with that additional uncertainty for the reference standard, then you also have then of course additional uncertainty for the measurement result. Uncertainties are very, very often visualized by the guard band principle and also used by the guard band principle. And we use it also now to show you a little bit more the risk that you are introducing with secondary standards. Before we do that, very quickly, a few words about the guard bands. They are commonly used. They are considering the expanded uncertainties, and they should be also as wide as the expanded uncertainties. They are used a lot in environmental analysis, in food analysis, in industrial analysis as well. Have also been suggested for pharmaceutical quality control a few years ago by Chris Burgess, for example, and also in our that is also by Chris Burgess here in our stimuli article in the Pharmacopeia Forum. So what do they do? So consider you have the lower limit for drug preparation, for example, or drug substance, and you have the upper limit for that for the pharmaceutical product. You have then here uncertainties for your measurement result that is normally symbolized by this kind of Gauss curve, and the uncertainty is then defining also the guard band. So you build in buffer zones into your specification zone, for example, and at the lower level, at the higher level here, and these buffer zones are as wide as the uncertainties to make up the acceptance zones. So why are they as wide as the uncertainties? Because if you have, let's say, a result here on the fringe of the acceptance zone to the rejection zone here, you have the result here, and the measurement that you have is always coming with it. There's always that uncertainty behind, but even with using then that guard band, you can be sure to a very high level, to more than 95% here in such a case, for example, that your result is still within the specification zone when you have the result here on the fringe of acceptance and rejection zones. So there is a high probability that your result is still within the specification zone. The same, of course, would be happening on the other side. So, and if you look then on the monograph situation there, where we have the conclusive combination of documentary standard and pharmacopoeia reference standard, then that uncertainty is not needed for the compendial purpose, and the acceptance zone is, in theory, of course, identical with the monograph limit. So it might be that users, when they verify the method into their laboratory, that they find for themselves during accuracy and precision studies for themselves an uncertainty that they want to put in as a guard band. But in theory, for a monograph, the acceptance zone and the specification zone would be the same. But we put in here uncertainties for a real reference standard that we gave the cover name, however, for surrogatine as surrogatine hydrochloride. And you see here the buffer zones put in the guard band. So that would be the situation for the monograph, for the conclusive combination here. But what is happening now when you are introducing a secondary standard that we have seen here in the measurement triangle was compared against the USP reference standard, then we are, by that additional measurement step, we are introducing additional measurement uncertainty to the value of the reference standard, but also, of course, later to the value of the reportable value. So we are adding uncertainty here into the game, and that would mean an extension of the guard band and would mean also a narrowing down of the acceptance zone, and that should really be considered in order to avoid unknown out of specification results, for example, because you would have, of course, a larger uncertainty. But if you still work with the old acceptance zone and say here and put it in here at the fringe of the old acceptance zone that we have seen on the slide before, then on this side here, the probability gets much higher to have a result outside of the specification zone, not just the acceptance zone, but of course the specification zone. And in theory, even the full uncertainty, because we are not in the conclusive area anymore, in the official combination of a compendial standard, the documentary and the reference standard. In theory, the full uncertainty of 0.96 would here apply then as a guard band. And what is additionally coming to that risk is that it is not fully possible for users to estimate an adequate narrowing of the acceptance zone, because the reference standards, compendial reference standards, are not providing uncertainty values that you could propagate into your result with a secondary standard. And why is that the case? Because the reference standards, the compendial reference standards, are made and set up for the official combination. That is their task, to provide the conclusive result on compliance by the combination of compendial standards, documentary method, and the reference standard. But it's not set up for propagating uncertainties or for assigning values to secondary standards. That's not the task of a reference standard, and therefore there's also no uncertainty provided to that material. And this is the risk that users of secondary standards to some extent are well, probably are aware and accepting, but very often when I talk to stakeholders, they are not aware of that statistical issue behind there, and what they should, however, in order to really know the risk and try things to mitigate that risk. For example, by narrowing down an acceptance zone, by whatever mean they want to use there as well. And the risk, of course, is can even be compounded depending on the method precision. When you are transferring a method into your laboratory, and it depends also how precisely you are working and transferring the method, how precisely you're weighing in, what analysts you are using, are they experienced, not so experienced, and then the method precision can also increase, of course. In the examples, we just used a reference, a method precision of a relative standard deviation of 0.2%, but there can also be higher precisions, of course, or let's not higher precisions, sorry, higher imprecisions here, so that we have a higher relative standard deviation. And you can see then how also the uncertainties change then when from using a pharmacopeal standard to a secondary standard. And here, at this point, you can come even to a uncertainty here that is already where you need to have the result really in the middle, and you have a very, very tiny acceptance zone only in order to make sure that you still fulfill the specifications there. So that is, of course, nothing that is new. That's inevitably the case. So because we have seen that already on the measurement hierarchy triangle there. So inevitably, the guard bands would widen with the transition from the pharmacopeal to the secondary standard, and they would also widen with increasing measurement standard deviation. So the conclusions that we have here is that the setup and the use of secondary standards, reference standards, in-house standards, working standards is of challenging complexity, sometimes not completely thought through by the users. And from our perspective, we can say that using pharmacopeia reference standards directly wherever that's possible saves you a lot of time and energy related to the inherently larger measurement uncertainty of the values that you assign to the secondary standards compared to the primary standards then. And of course then also consequently get the larger measurement uncertainties of the reportable values of the measurements you obtain for the product that is to be released. And it's also not so easy to adapt the acceptance and rejection zones by the transition from the primary standard to the secondary standard because of the special nature of compendium reference standards there, of course. But what should be clear is that it is appropriate to work with unchanged zones. So having an acceptance zone with a conclusive combination, with the official combination, let's say it like that, is one thing. But to use the same acceptance zone when going to the working standards, the in-house standards, that is an inappropriate kind of work, and it is definitely increasing the risk that we were talking about on the additional uncertainty here. And that is what we can definitely say. Establishing secondary standards or in-house standards or working standards, depends on how you want to call them, that always leads to more risk, and not only because of the uncertainty, but also because of what we said in the beginning, that only the pharmacopeia reference standard in combination with the documentary standard deliver your conclusive results. And with that, we are at the end of my presentation. I would like to make a few final remarks and acknowledgements. We have published what we were just discussing. We have basically published that also in an article that you can download from the website of Pharmaceutical Technology. So that's where we published that. And I want to thank my co-authors, Stephen, Ravi, and Doug from USP, and also especially Jane, who is one of our USP volunteers, one of these volunteers that are also balloting on the release of reference standards, as I explained to you on the slides before. And some other useful links there are, of course, the ones from USP, the direct one, the one about the reference standards there. And then, yeah, you can also have a look on the EMEA webinar playlist, series playlist on YouTube, where we have some other topics also discussed there in recorded videos from our webinars. And you can also download a white paper on the topic here, which is basically the gist of the article that's published here. So here we did a lot of calculations also of uncertainties. That's not done in the white paper, but it's giving you the gist of the article and also what we have discussed in this presentation here. And with that, I say thank you, and yes, let's stay connected and see what questions are coming up.
Thank you, Christian, for the very informative sharing. Next, I will take some time to give everyone an update from USP. And then in the meantime, please do keep your questions coming in. I can see a couple of questions in the Q&A already. Okay, I'll share my screen. All right, so next I will be giving you a very quick overview summary on the USP-NF Compendia update for the month of November 2021. Now, I will be giving you a very quick high-level summary of the new revision that were released in November. So over in the slide, okay, for your convenience, after this presentation, right, you are able to just click on the quick link to take a look at the notices as well as the respective monograph in detail if these are some of the related USP-NF monograph that you are working on in your laboratory. So first, for the baclofen injection, right, the update is that there will be a osmolarity acceptance criteria update. And then for the dextro injection, there is a change in official date. So it was supposed to be official in December 2021, right? And then it has been postponed to June 2022. And next, for magnesia tablet, there is a revision to remove identification B test, revert the assay from ion chromatography to titration. And then you will see there is a few of the magnesium-related substances for magnesium carbonate, hydroxide paste, oxide, trisilicate, as well as the milk of magnesia. The revision are pretty much similar, is to remove the identification B test, revert the assay from ion chromatography to titration, and revert the limit of calcium test from ion chromatography back to atomic absorption spectrometry. Next, for metformin hydrochloride extended release tablet, there will be an addition of dissolution test 25. For quetiapine extended release tablet, there is an addition of dissolution test 11. Starch TS, the revision is to provide alternative for the use of commercially available starch solution. For chlordiazepoxide hydrochloride as well as clindinium bromide capsule, there is an addition of dissolution test 2. And then lastly, for dexamethasone tablet, there is also an addition of dissolution test 2. So if these are monographs that you are working on a daily basis and you would like to understand more about the new revision, please feel free to click on the notice. All right, you can read more on the new revision in detail, and then this is the quick link to these respective monographs. And if you have further questions or any more inquiries, do not hesitate to reach out to your Apec representative. We have Rocky Chu representing and taking care of our customer in the Oceania region, Australia, New Zealand. and then myself taking care of our customers in Southeast Asia, and Shin Kim taking care of our activities in South Korea as well as in Japan. Now, upcoming activities in APEC in the year of 2022. January, very exciting. USP staff and external expert volunteers have collaborated to develop standards applicable for medicine excipients found in small molecules and biologics therapeutics. So this January, USP will be hosting a virtual training workshop on development and validation of bioassay, which is a workshop designed for individuals involved in regulations of advanced therapy within the APEC economy. So if you would like more information on this event, please do not hesitate to reach out to your respective representative. And of course, in January, do come back and join me with our next quality hour, the free live webinar. And then once the registration link will be available, we will send it out to you to join us for the 60 minutes free webinar. So with that, this is the end of my update, and we will move on to the question and answer, shall we? Right, so for the first question, this is a question regarding certified reference material. So there are two parts to the question. First, what are the difference between certified reference material and reference standard? I assume pharmacopoeia standard. And then the second part of the question is, does certified reference material can be replaced pharmacopoeia standard?
Yeah. Thanks, Stella. I also saw the questions already. There's also one remark, one comment in the chat about if we can kind of share the presentation. Of course, so from my side, that's completely possible. I can send you a PDF of that, and you can make it available to the participants. But let's come to the question now as well. So the first part of the question is, what are the differences between certified reference materials and reference standards? For that, we would have to go to the definition of the term certified reference material and reference material. There are two, and that is not something that is so far having a lot of relevance in the pharma area. Certified reference materials were introduced into the food industry, the environmental analysis industry, also the heavy industry when it came to checking iron ore on the content of iron in that ore, for example. So in that areas they were introduced first, and only slowly they get there. They get into the pharmaceutical area as well. But nonetheless, that is a very important question and a good question. And the international metrology vocabulary that you can download from the website of the BIPM, the International Office for Weights and Measures. In that international metrology vocabulary, there are the definitions for certified reference materials and for reference materials. And basically, reference standards, as they are in the pharmacopoeia, and it's also mentioned in our chapter 11, I think it's in the chapter 11, not in the general notices, but either in the general notices or in our chapter 11, it's mentioned that the reference standards fulfill the status of a reference material in the VIM, so in the metrology vocabulary. This is a material that needs to be stable enough for and needs to be fit for use for its assigned purpose, let's say it like that. And that is the case for our reference standards. They are for that specific purpose of the conclusive combination. They are fit for their purpose. That is what we have discussed in our, or that we have checked in our collaborative testing, for example. Certified reference materials are a little bit different. They also provide an uncertainty to the value of that they have set up So they are also having an uncertainty to the measured value, for example. So that you can use it in a kind of uncertainty calculation, like I have shown you on the slides as well. So there's the certified reference material, which is more or less, let's say, very sloppy. It's a reference material with an uncertainty, and that uncertainty is has been calculated by metrological traceability to, at the end, to the SI unit that is on the top of the measurement hierarchy. So that is the difference between a certified reference material and a reference material. And as I've said, reference standards from USP and from other compendia, they are on the level of a reference material. So that's the one thing. The second, exactly. So the second question, or part of the question, is: does a certified reference material can replace reference standards? Well, that's a different story. It depends on what you want to use that material for. When it is about getting a conclusive result, then it cannot replace the USP reference standard, even if it has an uncertainty in there, because it's not the official reference standard, and it's not the official combination of compendial standard or documentary standard and reference standard. So to that regard, it cannot replace, of course, the reference standard, the compendial one. That's all that I can say there. I don't know in which direction the question would go further. We can also discuss that offline when there is a special question that the person that put the question in there, if they have a specific purpose behind that question, we can go in more detail. But just from how the question is there, it cannot. A certified reference material cannot replace the reference standard for the conclusive result. That's not possible. You can use it like you use it. You use a secondary standard, for example. You can do then some work and thinking, what risk is it that I am accepting with using that certified reference material? But in the end, in last consequence, it cannot replace in order to deliver a conclusive result. Conclusive in relation to our general notices 580.
Okay, right. Thanks, Christian. And there is a second question. Okay, I'll read this out. How wide the range for secondary standard potency?
Now, where is that also still in the question?
Q&A tab.
In the Q&A? I can't see it there anymore. Yeah, can you? I only see the two questions that we just discussed. Can you repeat the question, please?
How wide the secondary standard potency? This was the question. So maybe the yeah.
How wide the secondary standard's potency or uncertainty?
Potency was the question.
Okay. Well, that's difficult to say. Normally, a reference standard should always be as pure as possible. That is especially true for the primary standard, because there, the primary standard, you assign a value by the mass balance approach. And what the mass balance approach is that you look on each impurity of the reference material, so everything that is not really the analyte that you are interested in, and you're subtracting that from 100%. And in the end, what's left over then is, of course, then the assay, the potency of that reference standard. And the more impurities would be in such a reference standard or reference material, the more error you can do in, of course, determining these impurities and subtracting from 100%. So that's for the primary standard. The secondary standard is then normally something that is compared to the primary standard by whatever procedure is used. So you have the reference point of the primary standard and then the comparison to that. So it's not really possible to say what purity it should have, because Well, I would also say there that secondary material should also be quite pure. It should definitely fulfill what is mentioned in the monograph, the specifications of the monograph. So it should be a material that is compliant to the monograph. That doesn't necessarily mean that when you have 98 to, let's say, 102% in the specification of the monograph, that 98 is okay. It could be okay, but it could also be, and that is also one of the risks of a reference secondary material, that there is, because you do normally a chromatographic comparison there, and it could be that you still have in that secondary standard an impurity, for example, that is eluting with the main peak, within the main peak, and then you would overestimate the purity of that material, because that peak of impurity would count also as purity in the, because the spectroscopic detection cannot distinguish between what it's seeing there. So that could also be a problem. So it must be a pure substance as well, and at least that peak for the analyte need to be really just the analyte, and you need to make sure that there is not an impurity underlying there as well. Yeah, so that's something that I can say, and it should it should fulfill, of course, the specifications of the monograph. But even 99% or 98% might not be enough when there is an impurity hidden maybe under the main peak.
Thank you, Christian. I hope that answers your question to the participant that submitted the questions. And then I do not see additional questions submitted to the portal right now. All right. So we are 3 minutes spare to 4 o'clock. I don't see any questions, so I will take this time to wrap up today's session. So thanks very much, Christian, for joining us today in your early morning. We really appreciate that. I hope to all audience that join us live today that you find this session helpful. It's a very brief, concise session. Please do not hesitate to reach out to your Apec representative if you like to discuss more in depth on the values of pharmacopoeia standards is related to your work at your laboratory or at your manufacturing facility. To all, please be assured that we are here to help answer any of the questions that you might have. And then we hope that you will keep in touch and see you next year in our January Quality Hour webinar. So after this session, you will actually be receiving an email to give us some feedback on today's Quality Hour webinar. So I hope that you will take some time to fill out the survey form, because your input is important to us to further improve the content of our webinar. So with that, I thank you for your time and participation. We really hope to see you again next year. And then I would like to take this opportunity that from all of us in USP, we wish you and your loved one a very advanced Merry Christmas, and then a successful New Year ahead. And then with that, I wish you a pleasant day ahead, and then please keep safe, and then take care. Thank you. Thank you, Christian.
Thanks. Bye-bye.
Bye.